Chapter 15, Segment 2, pages 476 through 483. Facial expressions are mediated by muscles, cranial nerves, and the central nervous system pathways. Facial muscles can be divided into two categories: superficial and facial muscles, which attach to facial skin. When these are contracted, they change the shape of the mouth, eyes, or nose, or they pull on their attachment to the skin. The frontalis wrinkles the forehead and raises the eyebrows. Deep facial muscles attach to the bone. These enable movements such as chewing and large movements of the face. Temporalis is a deep muscle located in the jaw. Human facial muscles are innervated by two cranial nerves. Facial nerve, 7, innervates the superficial muscles of facial expressions. The motor region of the trigeminal nerve, 5, innervates muscles that move the jaw. The main trunk of the facial nerve divides into upper and lower divisions after entering the face. These nerve fibers start in the brainstem, in the nucleus of the facial nerve. Facial feedback hypothesis suggests that sensory feedback from our facial expressions can affect our mood. In some studies done to test this hypothesis, a participant will place a pencil under their nose or between their teeth, and when asked how they feel, people who had formed smiles were found to be happier. People who receive Botox injections, which paralyzes muscles, experience emotions less intensely after their treatment. In schizophrenia and Parkinson's disease, chronic selective inhibition of facial musculature is a system that is present. Bell's palsy is a condition in which a virus infects the facial nerve and damages it, which results in the paralysis of facial muscles. This usually affects only one side, and it results in various symptoms such as drooping eyelids and mouth. No standard treatment exists, but most people recover on their own within a few weeks, and almost everyone is recovered by 6 months. Do distinct brain circuits mediate different emotions? Particular brain regions are involved in emotions, but the same regions often seem to be involved with many different emotions. Electrical stimulation of the brain can produce emotional effects. By electrically stimulating sites in the brain of awake, freely moving animals and observing their behaviors, you can study the neuroanatomy of emotions. Stimulations will have either a rewarding effect, aversive effect, or it may elicit a sequence of emotional behavior. The brain self-stimulation is the process in which animals will work to provide electrical stimulation to a particular brain site, presumably because the experience is rewarding. This can happen in humans too. Patients receiving the electrical stimulation in the brain region of the septum report feelings of pleasure or warmth, and in some cases, people reported that stimulating this region provoked sexual excitation. Many investigators have employed similar self-stimulation techniques to map the distribution of brain sites that produce self-stimulation responses. Animals will work to receive stimulation of many different subcortical regions, but stimulation of the cortex does not have a rewarding effect, as it seems. Medial forebrain bundle is a large tract that ascends from the midbrain through the hypothalamus that has a concentrated amount of effective stimulation sites for self-stimulation. This bundle of axons is characterized by widespread origins and innervates an extensive set of forebrain regions. One important target is the nucleus accumbens because dopaminergic stimulation of this site appears to be very pleasurable. One theory suggests that electrical stimulation taps into the circuits, leading more customary rewards. For example, the arrival of food to a hungry animal or water to a thirsty animal. Brain lesions affect emotions. Decorticate rage is the sudden and intense rage characterized by actions that lack clear direction. This is also known as shams rage. Dogs that had their cortex removed display decorticate rage by responding to routine handling with snarling and biting. This led us to believe that emotional behaviors of this type are organized at subcortical level, with the cerebral cortex normally providing inhibition of emotional responsiveness. James W. Papes proposed a subcortical circuit of emotion. He noticed an association between emotional changes and specific sites in brain damage. These interconnected regions became known as the Papes circuit and include memory bodies of the hypothalamus, the anterior thalamus, the cingulate cortex, the hippocampus, and the fornix. Paul MacLean, in 1949, proposed that the amygdala and various other regions also interact with the components of the circuit and suggested the entire system be called the limbic system. Reports of unusual emotional syndrome in primates with temporal lobe lesions provided early support for limbic system of emotion. Kluver-Bucy syndrome is a condition brought about by bilateral amygdala damage that is characterized by dramatic emotional changes, including reduction in fear and anxiety. While studying cortical mechanisms of perceptions, researchers removed large portions of temporal lobes in monkeys, and the animal's behaviors changed after the removal by becoming tame and showing no fear or aggression. They also showed strong oral tendencies such as eating various objects, including rocks. Frequent mounting behavior was also noticed and termed hypersexuality. Fear is mediated by circuitry that includes the amygdala. Animals tend to display the same behavior when fear is provoked. This similarity across many species may be the reason why we know more about the fear's neural circuitry compared to the other various emotions. Fear conditioning. A person or animal is presented with a stimulus such as light or sound, and it is paired with a short aversive stimulus such as a mild electrical shock. After a few pairings, the response to light or sound itself is the actual fear, which includes freezing and automatic signs such as cardiac and respiratory changes. These studies have provided us with a map of neural circuitry, and the amygdala is a key feature in the role of fear. The amygdala is located at the anterior medial portion of the temporal lobe, and is composed of approximately a dozen different nuclei, each with a distinct set of connections. Lesions to the entire amygdala appeared to abolish fear in monkeys, and lesioning just the central nucleus of the amygdala has the same effect. Prevents blood pressure from increasing and freezing behavior in response to conditioned fear stimulus. Information about fearful stimuli reaches the thalamus and can take two pathways to the amygdala. The low road is a direct projection from the hypothalamus to the amygdala that bypasses conscious processing and allows for immediate response to the stimuli. The high road, though, is the information flows through the sensory cortex, so this information can't be processed. It might be slower, but it's conscious and integrated with high-level cognitive processes such as memory. Contributions from prefrontal cortex and anterior cingulate are involved with another form of fear conditioning called observational fear learning. We can learn to fear something by observing signs of fear and pain in others without directly experiencing the fearful stimulus. Information about the fearful stimulus from several brain regions reaches the lateral part of the amygdala first, and evidence suggests that neurons here encode the association between specific stimuli and an aversive event, such as an electrical shock. The lateral amygdala triggers a network within, and this activates the central nucleus, which then transmits information to various brainstem centers to evoke three different aspects of emotional responses. Pathways through the central gray evoke emotional behaviors. Pathways through the lateral hypothalamus evoke autonomic responses, and pathways, the bed nucleus of the stria terminalis, evoke hormonal responses. Different types of stimuli activate slightly different pathways to and from the amygdala. For example, odors from an aggressive male will trigger different pathways than odors from a predator. Learned fears are hard to extinguish. During extinction training, activity in the prefrontal cortex, which extends to the amygdala, is necessary. If the prefrontal cortex is silenced during extinction training, the next day the animal will respond as though no extinction training took place the day before. Mice missing one of the two types of cannabinoid receptors also show a harder time learning their fear to a specific stimulus. This suggests that stimulating these receptors will normally extinguish learned fears. If this idea is correct, we may be able to develop cannabinoid drugs to treat phobias in humans. When humans are presented with a visual stimulus associated with fear or pain, blood flow to the amygdala increases, even when the conscious experience of the stimulus is not present. People suffering from temporal lobe seizures that include the amygdala commonly report that intense fear usually provokes a seizure. Likewise, electrical stimulation of temporal lobe sites may elicit fear in patients. SM is a patient with a rare genetic disorder that causes the accumulation of calcium deposits in the amygdala, and it eventually destroys the nuclei of both cerebral hemispheres. SM is unable to recognize facial expressions associated with fear in other people, but she can recognize other various facial emotional expressions. SM and other patients with this disorder have not experienced fear since the disease destroyed their amygdalas in late childhood, but they can be made afraid. When given only air with high concentrations of carbon dioxide, SM was flailing her hands, experiencing strong, panicky fear, and shouting, "Help me." So despite the central role of the amygdala with experiencing fear, the brain also has an alternative way of provoking fear and panicking us. One possibility of this is that the amygdala is important with regards to recognizing and responding to external threats, but the brain relies on other systems, possibly the brainstem, to detect and respond to signs of internal threats such as oxygen deprivation.